We've already learned about the concept of consolidation as it relates to memory. In fact, we've used the word in two different ways. We can refer to molecular consolidation or cellular consolidation. This is where cells, as a result of an LTP phenomenon, are going to build proteins, and the proteins are going to make synapses that were recently used stronger. That would be molecular consolidation. But then we also use the word to refer to system consolidation, where the hippocampal memories are going to be transferred out to long-term storage in the cortex. In this lesson, we'll encounter a concept called reconsolidation. It's getting a lot of attention now, and the concept applies to what happens when we reactivate a memory. Maybe you're recalling some autobiographical memory, some episodic memory, or you're just actually accessing your semantic memory. In either case, the argument here is that reactivated memories have to undergo another round of consolidation in order to retain the memory. And this has implications for the nature of memory. First, let's do some setup here. So here we have a graph here. The strength of the memory would be on the y-axis, and we can see sort of our two systems of memory, short-term memory system like the hippocampal system, and then the long-term memory system. So initially the hippocampal system will register some episodic event, for example. LTP will be happening in the hippocampus, but over time the hippocampal memories will be transferred out to the cortex, and we call that system consolidation. And we've learned that there are various things that can interfere with that process. Hippocampal lesions, for one. If you don't have a hippocampus, you cannot do the system consolidation. But we also learned that drugs can interfere with consolidation. Some of these drugs, like protein synthesis inhibitors, are going to block the late stage of LTP. And if you can't make the late stage of LTP in the hippocampus, then you're not going to have a chance of doing system consolidation to move the memory out to the cortex. We're going to add one more to the list, and that's electroconvulsive shock therapy. And it is well known that it has amnestic effects. That is, patients who undergo the treatment have difficulty remembering events just prior to the treatment, as though the ECS is blocking system consolidation. In fact, so we're going to use the electroconvulsive shock, I'll call it ECS, as a tool to study memory. Now, before we get to the concept of reconsolidation, the old view of memory seemed to be that once memories were sort of transferred out to the cortex, they were there in a relatively fixed state. And when you activated a memory, it didn't necessarily change the strength of representation out there in the cortex. You were just activating the memory, and then when you stopped, experiencing the memory, the cortical representation of that memory hadn't changed very much. So that was the old view. Well, in the early 2000s, a guy by the name of Nader is going to revive an older idea that memory becomes less stable and more vulnerable to change when we retrieve the memory. Right, so retrieval makes the synapses involved in the representation of that memory, it makes those synapses plastic again, modifiable again. So when we retrieve a memory, it becomes vulnerable then to procedures that weaken the memory. The memory must be reconsolidated to retain the memory. Now, using our graph, the idea then would be, we've already done system consolidation of some kind of memory. It's out here in long-term memory, but when we retrieve that memory, it becomes vulnerable, right, to procedures that can block consolidation-like processes. So we have to reconsolidate the memory in order to retain it in long-term memory. Now, Nader was working in the early 2000s, but the concept of sort of reconsolidation predates that. In fact, there were older experiments using ECS. And the idea is that consolidation blockers like ECS can weaken specific memories when given during retrieval, but not at other times. And here's the kind of logic of an experiment like this. So it'll be a rat study here, and we're going to see that the task that the rats are doing, they're going to be learning a fear memory, and to assess the level of fear, they're going to be measuring lick rate after the CS onset. So it'll be something like a tone and a shock, and the animal is in a cage that has some water, so it's probably going to be water deprived, right? So it's thirsty, and then it'll be licking its water bottle. And when the animal's licking a lot, it's quenching its thirst. But if something fearful comes along, it stops licking. So lick rate becomes a measure of fear, right? So here's the design of the study here. 4 groups of rats, and here's task acquisition. So you got the tone shock, right? On day 1. We'll just take a look at group 1. Then nothing's going to happen the next day, so they're not going to have a reminder of that learning experience, and they're just going to be tested the following day. So the test is going to be the CS alone, the tone alone, and then they're going to be measuring to what extent the rat is going to stop its licking when it hears the tone. If it has a good memory, right, of the pairing here, so tone shock, if it has a good memory, then it's going to stop licking. Let's take a look at group 1. Here we see that, in fact, they have a low, the lick rate is low here. In other words, the tone stopped their licking. Here they have a little indicator of the strength, the memory. So the lower the bar means the better the memory for the association between CS and US, tone and shock. So they were, in fact, afraid here when they heard the tone. They had retained the memory. Now look at group number 2. They're trained here, CS, US, but then immediately after training, they're given ECS. And we suspect that that's going to impair consolidation. Nothing happens the next day. They're tested the following day. Sure enough, when they are given the tone on the following day, they continue licking, right, compared to group 1. So the ECS seems to have impaired the consolidation of the memory, the fear memory. Take a look at groups 3 and 4, though. Interesting groups here to look at. Training on day 1, but then the following day, this group is reminded of the memory. They're just given the CS, so that's a cue. So it's as if they're going to be reactivating the memory, and in fact, they do show fear symptoms right on this day, and that indicates that they really are remembering the association between CS and US, the tone and the shock. But after they're reminded of it, right after they're given ECS, right? So this is not ECS after the training, it's ECS after a reminder of the memory, right? So they're reactivating the memory, they're retrieving the memory, and then they'll be tested. Take a look at group 3. It's as if the ECS now, kind of like ECS right after training, ECS after a reminder of the memory also interferes with retaining the memory, right? And compare that to group 4. You give the tone shock, and then a day later you do not remind them of the association, so you don't give the tone, you just give them ECS, test a day later, and the memory is still good. So comparing groups 3 and 4 shows that the influence of electroconvulsive shock, when given right after a reminder, it impairs the memory, as though a reactivated memory becomes vulnerable to procedures that can block consolidation. So what we would say here is that the ECS is blocking reconsolidation. Over here in group 2, the ECS was blocking the original consolidation. Okay, so let's use our familiar amygdala diagram to capture what's going on at a cellular level. So notice prior to training, right, so we have the tone pathway and the shock pathway converging on a target cell in the amygdala. Remember, you have to have convergence if you're going to set up this situation where you can associate one stimulus with another. And notice the shock pathway, that's a big synapse, so that's symbolic of a strong connection there, right? So that activity in the shock pathway will be sufficient to drive the target cell, to activate the target cell. But the tone pathway has a weak synapse, right? And this is why the tone originally does not generate a fear response, because of that weak connection. But because the two share a common target cell here, we can set up a situation where LTP will strengthen the weak synapse. And all you have to do is pair the tone and the shock. The shock pathway will be sufficient to activate the target cell, while activity in the tone pathway, so you'll get activity at that terminal, transmitter is released. And if the terminal takes part in driving the target cell, then you'll get LTP. Those are the conditions. You need target cell active while the terminal is active, right? Neurons that fire together wire together. So tone shock, tone shock, tone shock. What happens is LTP will be established, and you're going to need some protein synthesis, right, for the late phase of LTP. Now the tone pathway is strong enough to generate the fear response. That would be the conditioned response. Okay, so now let's take a look at the scenario, the groups that were presented in that study. So let's say we've trained the animal, tone, shock, tone, shock, tone, shock, but we give ECS right after the original training. Now here we're going to have the tone pathway as large here, but that's the early phase LTP. In order to get late phase LTP, you have to have protein synthesis, right? And it's possible that the ECS or protein synthesis inhibitors, what's going on here is you're preventing the late phase of LTP. So if you give ECS right after the training, then you're not going to allow the consolidation, molecular consolidation, to happen here. And so when you test on day 2, you're going to get no fear to the tone. So remember, they were given the training, they were given ECS right after the training. On day 1, nothing in that group, nothing happened to them. On day 2, they're tested, no fear, because that pathway did not get molecular consolidation. The ECS after training, and you can also use protein synthesis inhibitors, will block molecular consolidation. In the other group, we have tone shock training, and then on that next day, they just get ECS alone. But notice, between the training and that day, molecular consolidation was allowed to happen, right? So that synapse now becomes strong due to protein synthesis. And when you just do electroconvulsive shock at this stage, it does not weaken the synapse. It's already been consolidated. Molecular consolidation has taken place. So ECS is not going to affect that synapse. And so when you test a day later, they will show fear. So notice then the ECS has to be done right after training to block consolidation. Then you can block the memory. But if ECS is simply alone performed a day after the training, that won't have an effect on the memory. And take a look at this one down here. We have tone shock, tone shock, tone shock. And then on the next day, you give a reminder. So you activate the tone pathway and then give the ECS, right after the conditioned stimulus. Then it's as if this connection here becomes unstable again. It's vulnerable to a procedure that would block consolidation. And so we would say here that this is blocking reconsolidation, because the original consolidation was happening right after the training. So the ECS will block reconsolidation, which results then in a weaker synapse on the test day, and they'll get no fear. So the significance here is that when we retrieve a memory, and in this case the rat is just getting a reminder, right? So it's reactivating the memory because it hears the tone. Those synapses now have to be reconsolidated to maintain the memory. And if you come along and give a procedure that blocks that reconsolidation process, the synapse will be weakened compared to what it was prior to the reminder. You can use protein synthesis inhibitors as well here and get the same effect. Now, this effect of ECS sort of weakening a memory is different from, you'll recall, from the extinction process that we've learned about. You'll remember that in the typical extinction procedure, you just give the CS alone, right? And then the animal seems to be learning a second memory, another memory that inhibits the fear memory. And we think that there is this second memory because sometimes patients show recovery of the memory, especially when they're stressed out, or you give them the shock again, or it can just be spontaneous recovery. And so that suggests that the frontal cortex has to inhibit, right, the older memory, and anything that kind of interferes with the frontal cortex doing that can allow the old memory to resurface again in the amygdala circuitry. But when you do this procedure, when you block the reconsolidation, it's erasing the memory, right? So you don't get any spontaneous recovery. You don't get the return of the conditioned response with stress. You don't get reinstatement of the response after you give the shock alone. So this process, it's not a process where we're influencing some other memory and some inhibitory memory is coming along from the frontal cortex and inhibiting the fear memory. We are changing the very synapses that encoded the memory in the first place, right? So you're, in a sense, disrupting or weakening or erasing the original memory. Now here we'll come back to Karim Nader's work in the early 2000s. The ECS experiments were done decades before, but he had a similar sort of design. So again, it's a fear conditioning paradigm, and they're going to be measuring percent freezing. So when the fearful stimulus comes along, animals often just freeze. They stop moving. So here is the logic of the experiment. We got group 1, CSUS. Group 2, everybody's trained, right? But then the day later, the three groups have something different. Group 1, they're given a reminder, and they're given an injection of a fluid that has no other, it's cerebrospinal fluid, so it doesn't have any kind of impact on memory. It'd be like a saline injection. Group 2 had a reminder of the fear memory and then given a drug that blocks protein synthesis. And Group 3, on the next day, they're just given the injection of the protein synthesis inhibitor, and then they're tested a day later. So let's take a look. Group 1, they're given a reminder and then tested. They have a good memory of the fear. Group 2, they're given a reminder and then a protein synthesis inhibitor, and they've lost some of the memory of the fear, right? So reduced freezing, right? So it's as if the drug has blocked reconsolidation. Group 3, notice they didn't get a reminder, just the protein synthesis inhibitor, good memory on test day, right? So when you remind the animal, when the memory is reactivated, it becomes vulnerable to procedures that block reconsolidation, and a protein synthesis inhibitor blocks reconsolidation. You'll recall that LTP has the early phase, and that's going to trigger the cell to make proteins, and then you get molecular consolidation. So by giving a protein synthesis inhibitor here, they're blocking this process of reconsolidation. So an activated memory becomes vulnerable to modification. It has to be reconsolidated to be retained. And if we think about back to our sort of graph here, so, retrieving a memory, it has to undergo reconsolidation to retain it, which means if you retrieve and block reconsolidation, in a sense, you would be erasing the memory. Of course, it sets up a situation where memories could be erased, but also just changed and modified or distorted. So the phenomenon of reconsolidation applies that old memories can be weakened. And in a sense, we would experience that as forgetting, right? And we would model that, understand that biologically, that the strength of the memory was decreased because synapses, the LTP at those synapses, were reduced. Now, this is different from extinction, right, in which the new learning changes the response to an old stimulus, right? So this is not two memories being formed here. We are altering the original memory. Now, it does set up the possibility for distortion then, right? So every time we reactivate a memory, we might not be subject to electroconvulsive shock or have drugs injected into us to block protein synthesis, but we might be, after reactivating a memory, getting new information, right? And so in a period then during the reconsolidation phase, new information can be altering a memory. And we'll just kind of symbolize that with the little squiggle here. So every time we reactivate a memory, then it becomes vulnerable to additions of information. And that information might be accurate information, but it also could be inaccurate information, false information. So it raises the possibility for memory distortions. So because memory retrieval makes synapses plastic again, the pattern of reconsolidation can be changed by any new information being processed at the time of retrieval and reconsolidation. Now, this has an adaptive function. It makes memory updatable, but it also makes memory vulnerable to distortions after the storage of an original event. So, if we think about sort of historically how the understanding of memory has changed, in the old view is shown up here, where there's an event, we encode the event, we consolidate, and then we have kind of a fixed memory out there in the cortex, right? The consolidation would be here, the system consolidation from the hippocampal system out to the cortex. And the new view is shown down here, where, okay, there's an event, we encode it, we consolidate, and we have a memory out in the cortex, but every time we reactivate the memory, it becomes labile or modifiable or vulnerable to modifications. And we have to restabilize the memory, and this arrow would be the reconsolidation process that seems to involve protein synthesis. If you give protein synthesis inhibitors, right, you get disruption of the memory. You can give ECS here also and disrupt the memory. By disruption, we could say erasure of the memory, right? If you reactivate a memory and you have a new experience or you learn new information, then the memory will be updated, right? Maybe somebody tells you about something else that happened at some event that you're recalling, and so you can add that to your memory. So that's an adaptive function, but it does mean that our memories are vulnerable to information after the fact whenever we reactivate a memory. And let's just use an example with a rat trying to find food to show the value of having memory systems that are updatable, right? So let's imagine we have a situation where we got like these colored little plates here, and the rat is walking along and it finds food, right? Well, it has just crossed the green one, the black one, the blue, and the red one. Oh, at the red one it finds food. Well, it can make a memory of that event, right? And the memories are good because they help us predict what's going to happen in the future. So if the rat encounters the green plate again the next day or somewhere else, there's this situation, the rat encountering the green one can reactivate the memory. So the green plate becomes a cue now for the past experience. And now in the rat's brain, right, it can make a prediction because it has a memory of what happened in the past. And so it's going to predict that as it goes to the black and the blue and the red, it'll find food. And if it does find food, right, if it matches what the memory predicted, right, we can consolidate that, and the memory can be strengthened, right? So that's good. The rat has now a good memory for this pattern, and it's useful. It helps to predict where the food is. So now imagine on another day, so the rat finds the green one, goes the black one, the red one, or the blue one, and then the red one, and there's no food there. Now there's some new set of plates to walk on, right? So that's a mismatch. Right, it was expecting food here because the previous memory said that there was going to be food there. Now there's a mismatch, and that's going to stimulate the rat's brain, right, to explore here. And hey, where's the food, right? So it might go left. Oh, if it goes right, it encounters a blue and a red one. There's the food. And so the idea is that this new situation where the food was not where it was supposed to be, according to the previous memory, that's going to require that we update the rat's memory. Right? So we don't want to just keep memories in a fixed state forever. We want to be able to modify them and update them in the service of surviving, right? So the mismatch here triggers the rat's brain to seek out and explore and learn the significance of the change. And the hippocampal system then will be able to consolidate new information and to update the rat's memory. And that is clearly a useful function, to have a memory system that is updatable.